Here in chapter 3, we are going to be learning about cells, which is going to be the basic unit of all future discussions. So first of all, the functions of the cell and why we need them, starting with the parts of the cell. So all cells have a cell membrane or plasma membrane. We talked about this a little bit in chapter 2 with those phospholipids, but this is going to be the outer boundary that allows interaction with the external environment. So separates the inside and outside of our cells. Some cells also have a nucleus where the DNA is stored that directs the cell activity. As we'll talk about though in a minute, not all cells have a nucleus. But when we're talking about anatomy and physiology, all of our cells, except for red blood cells, do have a nucleus. And then they also have a cytoplasm, which is kind of a gel-like fluid inside of the cell between the membrane of the cell and the nucleus. And that's where the organelles like the mitochondria and that type of thing, as well as what are called ribosomes that help with protein production, would be found. Now, some of the general functions of cells is getting and using energy. So that metabolism, making molecules, so being able to make DNA or make proteins or carbs. It's also important to communicate with what's going on outside of the cell as well as inside. And then it's involved in reproduction and inheriting. So that's our genetic information, our DNA. Here's a look at a typical animal cell, or in this case, human cell. Again, this is showing the membrane, so that phospholipid bilayer where those phospholipids, the head's pointing out and the tail's coming together to create kind of a barrier from the inside and outside. We have the nucleus, which is where the DNA is stored. Outside of the nucleus, we have the endoplasmic reticulum, which is just a series of channels. Some parts of this is smooth, called the smooth ER. Some have small ribosomes attached. This is the rough endoplasmic reticulum. These are synthesizing molecules, so our proteins, our carbs, and lipids get made here. Our mitochondria, kind of a funny looking organelle, that's where our energy, our ATP, the majority of it is made. And the processing or kind of shipping UPS center of the cell is called the Golgi. Again, a series of flattened sacs. There are vesicles coming in, so it's bringing in things from the ER. They get modified and sorted, and then they get packaged and distributed to wherever they need to be. So in this case, the product is being released outside of the cell through a process called exocytosis. In addition, we have some other smaller vesicles, lysosomes, which are really important to our immune system as far as breaking down like bacteria and foreign material, but also just kind of the trash can recycling center. And then our peroxisomes, that's the detox center. So toxins that get made during metabolism, they get broken down. So how we see cells. So cells are microscopic, so we need a microscope to see them. A little bit about the different types of microscopes. So light microscopes, this goes up to about 1,000 times the actual size. This is good for looking at various tissues and doing biopsies and looking at different cells. Now, it does require some stains for most of these, so you can actually see like the nucleus or organelles within the cell. If we need to go to a higher magnification, we need more detailed specific things inside the cell or on the surface, electron microscopes get used. And there are two different types. Scanning electron microscopes give a three-dimensional surface view, so looking at things like receptors or molecules outside of the cell, microvilli or cilia that get attached. But if we want to look at detail inside of the cell, the transmission would show that high magnification of internal structures. Now again, back to our plasma membrane. Again, as far as functions, as we mentioned, this is the boundary, separates the inside and outside or intracellular versus extracellular environment. So it encloses and supports the contents of our cells. It also attaches to our extracellular environment. So if we're forming tissues where cells are coming together, that membrane helps attach. It's involved in recognizing and communicating with other cells and moves things in and out, regulates what's coming in, kind of a gatekeeper in and out of those cells. Now, across these membranes, we can have what's called a membrane potential. And this is an electric charge difference that's across that membrane, meaning the inside has a different charge than the outside of the cell. If that's the case, then it has a membrane potential. And for our cells, there are more positively charged ions outside of the membrane, giving it a positive charge outside. And then there are more negatively charged, especially like our DNA and RNA that have negative phosphate groups on them. So that makes the inside of our cells negatively charged. So our membrane, again, shown here, the main molecule that makes them up are those phospholipids that we've mentioned. Again, hydrophilic head, two tails sticking out. They form this bilayer, two layers of these where their tails come together and the heads are on the outsides. This creates a barrier that most things can't cross through. Now, in addition to those lipids, there are also different proteins. So some that go just on the internal surface, some that go all the way across on both sides of the membrane. We also can have some carbohydrate chains that stick out there, and they form what is called glycolipids or glycoproteins, just meaning they have sugars attached to the lipid or the protein on the surface. And a lot of times those combine to form what's called the glycoclax, which help them attach to other cells, also helps with our immune system to recognize the cell as being a self cell. Now these membrane lipids, again, those phospholipids form that bilayer with the heads facing the water, the tails facing each other, where it creates that hydrophobic region. In addition, cholesterol, which is also hydrophobic, doesn't like water. These are just small molecules, just shown here in yellow. Of course, cholesterol gets a bad rap because too much definitely hardens our arteries. It's bad for us, but we do need a little bit because it helps with fluidity. So these membranes are constantly moving. They don't sit still. So that cholesterol helps add some stability to the membrane. Now we do the membranes follow what's called a fluid mosaic model because they are fluid in nature, again, just meaning they're moving. This picture diagram, of course, is just setting still, but our membranes, all of these parts are constantly moving around, distributing molecules. They can reassemble if they get damaged, and again, they can kind of come or fuse together. Now, the proteins that are in them, we have integral proteins. These extend into the membrane and extend only from one surface to the other. So they go all the way across the membranes, both bilayers, and can form channels through the membrane. So they're part outside, part inside, crosses the entire membrane. Now, peripheral proteins, as the name implies, is just on the periphery. So it's just that either the inner or outer surface, not all the way across. There are several different types of proteins that can be on our membranes. One of them is referred to as marker molecules. And these are some of those glycoproteins or glycolipids that have that sugary or carbohydrate layer attached to them. And these mark or identify the cells. So this helps our immune system recognize self versus foreign cells by these glycoproteins or marker molecules that are embedded in the membrane. And they also help with intracellular communication, so communicating with what's going on inside of the cell. Attachment proteins, as the name implies, help cells attach to the outer surface, so parts of that extracellular fluid or extracellular part of the cell. Some integral proteins that help with attachment are called cadherins. These attach cells to other cells. So if we're making a tissue and we're joining cells together, cadherins are proteins that would kind of act like that glue. And then integrins pair the membrane to extracellular molecules. Like we see here, this attachment protein here. So these would be integrins because they're attaching not to another cell, but to something outside of that cell. Transport proteins we'll talk more about. As the name implies, though, they're literally transporting or moving molecules. Again, one of the functions of membranes is to control what's coming in and out, and this is the main way that they do that. Now, there are a few different types. So there are channel proteins, and these essentially are just a passageway or a hole through the membrane. We also have carriers or transporters. They are physically changed. They attach to a molecule and change shape to deliver it inside or outside of the cell. And then sometimes we need energy for these. So we have ATP pumps. Now, regardless of the type of transport protein, they're all specific, meaning they only allow specific molecules through, like a specific ion or a specific molecule like glucose. It's not just a general purpose molecule that transports anything. There can also be competition if there's molecules of similar shape. So sometimes drugs work by blocking or activating some of these proteins to alter what's coming in and out of cells. And they can become saturated, just meaning, especially with the carrier proteins, if we have a lot of molecule outside, but only one transporter, then it's going to get saturated. There's not going to be a lot of movement because there's not enough transporters to move everything. Now, a little bit more about these transport proteins. So the ion channels, so these are channels, again, holes kind of through the membrane that let ions through. Some of them are called leak, meaning they're not gated. So they're just kind of always open. Now they're still selective. They're only letting certain molecules through, but they're just moving with their concentration gradient, meaning they're going from a high to low concentration, and they're always open. Now, gated channels, though, have a gate on them, as it implies, so they can be open or closed based on a stimulus, and that stimulus can be different. There are ligand-gated ion channels. So these open based on a molecule called a ligand that physically binds to the protein or glycoprotein that activates it. And then voltage-gated, back to that membrane potential where we have opposite charges, the outside being more positive, inside more negative. So if that changes, then that can open those channels. And then just kind of to relate this, cystic fibrosis is actually a genetic disorder where chloride ion channels don't work. So chloride ions can't move across the membrane, and that creates the really thick, viscous secretions and mucus that lines their lungs and can cause problems with digestion. And this is just showing those different ones. Again, these are leak channels. They're always open. There's no gate, so things just come in and out based on going from a high to low concentration. Ligand gated means there's a molecule, so a binding site, kind of like our enzymes. So something specifically attaches, and then it will open up. So only when that ligand is present does that gate open. Now, carrier proteins, also called transporters, these are going to physically bind, but then they carry the molecule across. So the molecule they're carrying comes in, again, it has a specific shape, it fits into this site. Once it attaches, that causes the protein to change its shape, that three-dimensional tertiary structure. It changes that shape and it releases the molecule on the opposite side of the membrane. Now, the direction that they go give us some different terms. So uniporters, that means it's carrying one molecule or ion. And symporters move two ions or molecules, but they're going in the same direction. So this is called co-transport. So it may be bringing in hydrogen ions and glucose, but both of those are coming into the cell. So they're going the same direction. Then antiporters are moving two different ions or molecules, but in opposite directions. So sodium, potassium. So one is coming in, sodium comes in, potassium goes out. So they're moving in opposite directions. And then if they need energy, they're called pumps. So it means they only function if energy and ATP is provided. Now, receptors are another protein that's on the membrane. Receptors are important for receiving and communicating with what's going on outside. So again, these are proteins or glycoproteins, but they have a site on the outer surface and that attaches to a specific signal, like a hormone, for example, an insulin receptor. Insulin would bind to, again, a specific shape, kind of like our enzymes. It fits into that receptor, and then it activates the receptor, and that communicates and starts a process inside of the cell. And then sometimes enzymes are directly placed on our membrane, so they can be membrane proteins. So again, enzymes are proteins that speed up reactions. Typically, they're just inside of the cell or inside of an organelle, but sometimes the enzyme is placed in the plasma membrane or in a membrane in order for it to work.